I represent the speakers, so I'm really proud to present our colleague Laura Silvestri, and she's from San Raffaele Scientific Institute and Ospedale in Milan. A research activity focused on iron and metabolic-related diseases. I think that hepcidin era and liver BMP SMAT signaling pathway are one of the topics of interest, but we will see that we have to learn a lot from you. So thank you very much for taking this talk.
Thank you very much. Thank you very much, Beatrice, and good afternoon, everyone. So it's my pleasure to be here and to share with you some findings about the crosstalk between iron and erythropoiesis, and with a special focus on inherited forms of iron-loading anemias. So can you see the presentation?
Perfectly, yeah.
Okay, let me select, yes. Okay, can you see the pointer?
Yes, indeed.
Okay, that's great. So this is the outline of the presentation. In the first part of the talk, I will discuss the functional and reciprocal crosstalk between iron and erythropoiesis. Then I will discuss a little bit more about the iron-loading anemias, and in particular be focused on the definition and the pathogenetic mechanism that causes iron overload in conditions of ineffective erythropoiesis. And then I will focus on the new therapeutic approaches with the special using, I would say, as a paradigmatic example, the new findings that we have on beta thalassemia. So, as you may know, erythropoiesis is a multi-step and complex process that can be divided in early phase, in late phase or terminal erythropoiesis, and in a maturation of reticulocytes towards mature RBCs. So as you can imagine, we have several cytokines and proteins that are involved in the complex maturation process that from hematopoietic stem cells originates mature RBCs. But today I will focus only on two of them, erythropoietin and iron. So erythropoietin is a growth factor that exerts its function mainly in CFUE, so in the early phase and in terminal erythropoiesis. And it controls the proliferation and differentiation of erythroid precursors. Erythropoietin is produced by the kidney and is under the control of the hypoxia-inducible factor 2 alpha. So in conditions of reduced body oxygen tension, such as in conditions of anemia, if 2 alpha gets stabilized and increase the production of erythropoietin. So as you can imagine, hypoxia and body oxygen tension is regulated by RBC's output from erythropoiesis. So meaning that the amount of RBCs that we release in the circulation indirectly controlled the stability of E2 alpha and thus hypoproduction. So when RBCs are among normal values, oxygen tension is fine, we don't have hypoxia if 2 alpha gets stabilized and hypoproduction is reduced. The opposite occurs when RBCs are decreased, such as in case of anemia or in case of iron-loading anemias. So as you can imagine, iron plays a crucial role in erythropoiesis and RBC's maturation. And you can think about the amount of hemoglobin that contributes to RBCs. We know that about 90% of hemoglobin contributes to RBC's dry weight. And hemoglobin is made by heme, that means prosthetic groups and iron, and by globin chains. So iron is a crucial component of hemoglobin and is a crucial element that controls erythropoiesis, erythroid cell production and erythroid cell differentiation. a huge amount of iron is consumed by erythropoiesis. We are talking about 25 milligrams of iron per day in basal state condition. You should consider that only 1–2 milligrams per day of dietary iron is absorbed through duodenal enterocytes. So that means that this huge amount of iron should be provided by the degradation of senescent RBCs by reticuloendothelial macrophages. So given the crucial role of iron in the production of hemoglobin and RBCs, the erythropoietic activity can directly control the amount of iron that enters in the circulation. So in the first part of the talk, I would mainly focus on how does iron modulate erythropoietic activity. So iron is taken up by erythroid cells mainly in the form of the ferric transferrin. And in erythroid cells, there are two receptors that can bind the ferric transferrin. One is a transferrin receptor 1 or CD71, whose function is to provide iron to erythroid cells. And the other one is a transferrin receptor 2 that has mainly regulatory function. Indeed, it can bind a EPO receptor, thus regulating the EPO receptor signaling. So if we focus on terminal erythropoiesis, that means the process that from pro-erythroblast produced RBCs, the main form of iron that is taken up by erythroid precursor is the ferric transferrin. So the iron is released by nurse macrophages that plays a crucial role in providing not only iron but also growth factors and cytokines to erythroid cells in order to allow the maturation towards RBCs. Nurse macrophages are the central macrophages of the erythroblastic island. So in erythroid cells, TFR1, the interaction between the ferric transferrin and TFR1 triggers the ligand and receptor internalization. And in the acidic endosomes, iron gets released from transferrin and transferrin receptor. It gets reduced by STIP3 and then released into the cytosol by divalent metal transporter 1. And if we look more in detail on how TFR1, STIP3 and the NT1 that contributes to form the machinery that provide iron to erythroid cells gets expressed, we can see that they are clearly highly expressed in pro-erythro and basophilic erythroblast, but they get downregulated according to erythroid cell differentiation, meaning that iron uptake is crucial in the first phase of terminal erythropoiesis. Once in the cytosol, iron should be stored into ferritin nanocages thanks to the chaperone activity of PCBP1 and PCBP2. And indeed, both ferritin H, ferritin L, and PCBP1 and 2 are highly expressed in accordance with TFR1, STIP3, and DMT1. So in the first phase of terminal erythropoiesis, erythroid cells try to uptake more iron as they can and to store it into ferritin. At this point, iron should be released back in the cytosol, and this occurs through a process named ferritinophagy that involves the precise lysosomal degradation of ferritin nanocages thanks to the cargo receptor NCOE4, which is more expressed during orthochromatic erythroblasts in terminal erythropoiesis. So in the first phase of terminal erythropoiesis, iron should be taken up and stored into ferritin, but as soon as the cells differentiate towards orthochromatic erythroblast, then iron should be released back into the cytosol. So what is the fate of iron once it reaches the cytosol? It should be transported into the mitochondria. And the mitochondria plays a crucial role in erythroid cells, since in the mitochondria iron is used for two main biochemical processes. The first one is the iron sulfur cluster biosynthesis, and the second one is the heme biosynthesis. And we will see from this complex scheme that I'm going to explain a little bit more that both the iron sulfur cluster machinery and the heme biosynthetic pathway are closely related. So iron in the cytosol enters into the mitochondria through mitoferrin 1. And in the mitochondria, it can be used to synthesize iron sulfur clusters. These iron sulfur clusters are prosthetic groups that regulate the activity of enzymes that are important, for example, for the OXOS and Krebs cycle, but that are also important for heme biosynthesis. And indeed, ferrochelatase rely on, the activity of ferrochelatase relies on its ability to bind and to be regulated by an iron sulfur clusters. However, iron sulfur clusters exert their function not only in the mitochondria but also in the cytosol. Indeed, they are, once synthesized, they are released into the cytosol through ABCB7, and here in the cytosol, they regulate the RNA binding activity of iron regulatory protein 1 and iron regulatory protein 2. These two proteins are crucial because they sense the amount of iron that the cells has stored, and they regulate the uptake, the export, and the utilization of iron according to the iron availability. And the cells senses its iron availability thanks to the iron sulfur cluster synthesis. So, for example, when iron is sufficient and it's enough to fuel the iron sulfur cluster machinery, the RNA binding activity of IRP1 and IRP2 is blocked, and then the cell decreases the iron uptake because it has enough iron, but it increases its export, but also the utilization. And one enzyme that is regulated by this process is ALA synthase 2, which is the first and rate-limiting enzyme in heme biosynthesis. So ALA synthase 2 activity and in general heme biosynthesis is closely related to the amount and the activity of the OXOS and Krebs cycle, because succinyl-CoA, which is one intermediate of the OXPHOS and the Krebs cycle, the amount of succinyl-CoA is mediated by the activity of the enzyme that are regulated by the iron sulfur cluster produced here. But also ferrochelatase activity is under the control of iron sulfur cluster. So heme is produced and is synthesized both in the mitochondria, but the biosynthetic pathways takes place also in the cytosol, but I don't want to go too much into detail on this. Once synthesized in the mitochondria, heme is released in the cytosol through the Feli leukemia virus receptor type C. And here in the cytosol, heme regulates the synthesis of alpha and globin chain in order to produce hemoglobin. This cross correlation between heme concentration and alpha and globin synthesis, alpha and beta globin chain synthesis should be preserved, because in the presence of less heme but enough and an increased amount of alpha and globin chain, we can have an impairment of the activity of alpha and globin chain that can be, somehow sequestered into aggregosomes that damage the red blood cells. So the amount of heme coordinate in a precise way the amount of alpha and globin chain that should be synthesized in order to synthesize hemoglobin in a precise stoichiometric relationship and ratio. So what happens when iron is reduced in erythroid cells? We have, of course, less iron that can enter into the mitochondria. So we reduce the synthesis of iron sulfur clusters, and this affects the mitochondria in several ways. First, it affects the activity of the OXOX and Krebs cycle, and it reduces the amount of succinyl-CoA that is produced. And second, it impairs the activity of ferrochelatase, which is the last enzyme in heme biosynthesis. In addition, we have less iron sulfur cluster that can be released in the cytosol. Thus, IRP1 and IRP2 are mainly in their RNA binding activity. That means that the cells are sensing that it has low iron concentration. And for this reason, the cell increases the uptake of iron, decrease the export, and decrease the utilization of iron. And the main enzyme in erythroid cells that is affected by a decreased iron sulfur cluster synthesis is ALA synthase 2. So ALA synthase 2 production is decreased because its synthesis is blocked by IRP1 and IRP2, following the reduced amount of iron sulfur cluster that is produced by the mitochondria. So overall, we have less heme that is produced by the mitochondria, less heme that is released in the circulation, and less hemoglobin that is produced, and we undergo anemia. So not only transferrin receptor 1, but also transferrin receptor 2 plays a crucial role in the regulation of erythroid function. Transferrin receptor 2 is highly homologous to transferrin receptor 1, but a difference with transferrin receptor 1 whose function is to uptake to trigger iron entry into the cells. Transferrin receptor 2 has mainly regulatory function. As transferrin receptor 1, also transferrin receptor 2 can bind the ferric transferrin, but with a lower affinity. And in particular, in erythroid cells, transferrin receptor 2 interacts with EPO receptor and regulates the EPO receptor sensitivity of the erythroid cells. In particular, as you can see here, transferrin receptor 2 is highly expressed in proerythro, and its expression is reduced towards differentiation. And this expression is superimposable to the expression of EPO receptor, thus suggesting that they interact functionally. Transferrin receptor 2 plasma membrane localization is regulated by the amount of circulating iron. So in particular, when the ferric transferrin is reduced, such as in conditions of iron deficiency or when we have a fluctuation of circulating iron, we have a decrease in TFR2 on the cell surface. So EPO receptor is free to interact with EPO, and the EPO receptor signaling is acutely increased. So how does the erythroblast adapt its function according to reduced iron availability? This is just a schema of what I previously presented. So in erythroid cells where iron is reduced, the iron sulfur cluster synthesis and heme biosynthesis are both decreased. So hemoglobin synthesis is reduced, but also RBC's production is decreased. The body, and in particular the kidney, is sensing a conditions of hypoxia. So it stabilizes HIF2 alpha, that is the transcription factor that increases the production of EPO by the kidney. However, in a conditions of persistent hypoxia that is combined by iron deficiency, the decreased amount of iron in the kidney decreases the stability of HIF2 alpha in order to compensate and avoid an uncontrolled activation of erythropoietin that in a conditions of iron deficiency can push too much erythropoiesis, thus consuming the reduced availability of iron that can be used for other biological processes. This is a mechanism that the body has, I would say, selected and developed in order to attenuate a upregulation of EPO production in a conditions in which iron is not available to sustain a proper production of mature RBCs. So I've shown how does iron modulate erythropoiesis, but what happens to erythropoiesis and how erythropoiesis shape body iron homeostasis. Erythropoiesis can modulate body iron metabolism by targeting hepcidin. So what is hepcidin? Hepcidin is the master regulator of iron metabolism that is produced by hepatocytes and is released as a secreted hormone into the circulation. The function of hepcidin is to prevent iron recycling and to inhibit intestinal iron absorption by binding and blocking the sole iron exporter ferroportin. So in the presence of hepcidin, when hepcidin rises for several reasons, ferroportin is removed from the cell surface and iron remains trapped inside the cells, in reticuloendothelial macrophages and in duodenal enterocytes. In this way, an increased hepcidin production reduces the entry of iron into the bloodstream. I just want to remind you that reticuloendothelial macrophages, or mainly splenic macrophages, plays a crucial role in recycling iron for erythropoiesis. Indeed, only 1 to 2 milligram of iron is absorbed by dietary iron, whereas erythropoiesis needs 25 milligram of iron per day for a proper hemoglobin and RBCs production. This means that iron should be recycled, and splenic macrophages plays a crucial role since they phagocyte and degrades senescent RBCs, thus providing iron back to the circulation for new erythropoiesis. So, hepcidin production and hepcidin expression can be reduced by several physiological stimuli. For example, if body iron concentration are reduced, hepcidin should be inhibited, but also in conditions of expanded erythropoiesis, hepcidin gets downregulated in order to stabilize the ferroportin on the cell surface and increase the amount of iron that enters into the bloodstream. This iron is used to fuel erythroid cells in order to allow them to produce an adequate amount of hemoglobin and to mature towards RBCs. And if we want to investigate more in detail, what are the mechanisms by which expanded erythropoiesis decreases and downregulates hepcidin production, I have prepared a scheme, a simplified scheme that illustrates the main players regulated by increased erythropoiesis and that allow hepcidin inhibition in conditions of expanded erythropoiesis. So in conditions of, for example, of decreased RBCs, such as in bleeding conditions or in severe iron deficiency, the kidney started to synthesize erythropoietin. The erythropoietin stimulates erythroid cells proliferation and differentiation. And in particular, among all the proteins that are stimulated by the EPO-EPO signaling, there is one protein that is named erythroferron, that is a secreted and a soluble protein that is produced by erythroid cells in response to EPO and is released into the circulation. This protein is named erythroferron or ERF, and it's an inhibitor of hepcidin expression. Thus, in conditions of expanded erythropoiesis, erythroferron downregulates hepcidin expression in the liver. This is an example of the crosstalk between two different physiological mechanisms and organs, erythropoiesis and liver and iron metabolism. However, in condition of persistent hypoxia, we have other tissues and cells that contribute to inhibit hepcidin. First one, hepatocytes released again a soluble factor named FGL1 that as erythroferron downregulates hepcidin with the same similar mechanism. On the other side, we have cells such as macrophages, platelets, and endothelial cells that release PDGF-BB that inhibit hepcidin through a different mechanism of action. All these factors, soluble factors, contribute to increase iron entry into the circulation because, as I mentioned before, in conditions of decreased hepcidin, ferroportin is stabilized, so we have more iron that enters into the bloodstream. This iron is used for expanded erythropoiesis to synthesize heme, produce mature RBCs, and to correct the hypoxia, the original hypoxia. However, there are pathological conditions in which the constitutive and expanded erythropoiesis, for example, due to ineffective erythropoiesis, causes a chronic hepcidin inhibition mediated mainly by the factors I described before, causing a chronic and persistent stabilization of ferroportin on the cell surface and an uncontrolled iron entry into the bloodstream. The buffering capacity of transferrin is overcome by this huge amount of iron that enters into the circulation, and so a toxic form of iron starts to appear. This toxic form is named NTBI, that means non-transferrin bound iron. That is a very toxic form of iron because it accumulates in several organs and tissues such as the liver, pancreas, and heart in an uncontrolled way, causing organ failure. However, NTBI is harmful not only for these organs, but also for ineffective erythropoiesis. And an increase in NTBI can further worsen the original ineffective erythropoiesis. So there are a group of disorders that are named iron-loading anemias that can be inherited or acquired that are characterized by ineffective erythropoiesis. So the definition of ineffective erythropoiesis is this one. Ineffective erythropoiesis is characterized by a reduced maturation towards RBCs due to an increased apoptosis of erythroid precursors caused by the inherited or the acquired defects. The anemia that originates in ineffective erythropoiesis stimulates erythropoietin production. Erythropoietin is a growth factor that stimulates proliferation that is preserved in conditions of ineffective erythropoiesis. It stimulates also maturation of erythroid cells towards RBCs, but in the presence of the genetic defects, this maturation is impaired, so the cells die. So in conditions of ineffective erythropoiesis, we have an imbalance between proliferation and maturation. That is basically the opposite in conditions of normal erythropoiesis. So ineffective erythropoiesis is characterized by low hepcidin levels, as I have shown to you before in the previous slide, that causes excessive iron absorption and secondary iron overload. And we can distinguish among two different forms, the inherited one and the acquired. But for sake of time today, I will focus mainly on inherited iron-loading anemias, and in particular, I will describe the pathogenetic mechanism that characterized sideroblastic anemia and beta thalassemia. So starting from sideroblastic anemia, this is the slide I have already shown that describe the mechanism by which the mitochondria synthesize iron sulfur cluster and heme. Interestingly, sideroblastic anemia, which is a genetically heterogeneous disorder, can be caused by mutation in ABCB7, which is the iron sulfur cluster exporter, or mutations in ALA synthase 2, which is the rate-limiting enzymes in heme biosynthesis. And mutations in ABCB7 and ALA synthase 2 characterize an X-linked form of sideroblastic anemia. However, we can have also an autosomal recessive sideroblastic anemia, which is characterized by mutations in the glycine importer SLC2538. And I want just to remind you that glycine is a component, is a crucial component in the first step of heme biosynthesis. But it's also caused by mutations in genes that are important and crucial in the modulation of the iron sulfur cluster machinery, such as glutaredoxin 5 and the chaperones HSPA9 and HSCB. We have other forms of sideroblastic anemia, such as the autosomal DOM-recessive sideroblastic anemia, characterized also by myopathy and lactic acidosis. And in this case, the mutated genes are PUS1, LARS2, and YARS2. Or we have a maternal sideroblastic anemia that is characterized by large mitochondrial DNA deletion. However, the common features of all these sideroblastic anemias are the presence of ring sideroblasts. Ring sideroblasts are these blue spots that surround the nuclei, and these blue spots represent iron-loaded mitochondria. So, but how does iron get iron-loaded in sideroblastic anemia? The mechanisms are several. First of all, and here the X indicate the genes that are mutated in sideroblastic anemia. First of all, we have an impaired production of iron sulfur clusters. And as I mentioned previously, iron sulfur clusters are released in the cytosol to tell the cells that they have enough iron. So less iron sulfur cluster in the cytosol mean that we have less iron in the mitochondria. So the cells stabilized IRP1 and IRP2 for their RNA binding activity and start to increase the iron uptake and to decrease the export, and also to decrease the utilization. And indeed, in this condition, allosynthetase 2 synthesis is downregulated. So even though we have a genetic defect that affects the iron sulfur cluster machinery, the final readout is a decreased heme biosynthesis because of a direct downregulation of allosynthetase 2 and a decreased activity of ferrochelatase that, as I mentioned before, rely on iron sulfur cluster prosthetic group. So we have a decrease in iron sulfur cluster. We release a false signal of iron deficiency. That means that the cells try to uptake more iron. In this condition, mitoferrin-1 is upregulated, and the mechanism by which mitoferrin-1 gets increased is still unknown. And as I mentioned previously, mitoferrin-1 mediates iron uptake into the mitochondria. So more mitoferrin-1 on the mitochondrial membrane, more iron inside the mitochondria. Mitochondria gets iron loaded. The oxidative phosphorylation and Krebs cycle are affected because I have less heme and less iron sulfur cluster that are both important for the activity of these enzymes. I have less succinyl-CoA and less energy. The increased uptake of iron by the mitochondria increase reactive oxygen species production. It impairs mitochondrial function. I have a reduced heme biosynthesis and, of course, a reduced hemoglobin production, and anemia occurs. In beta thalassemia, ineffective erythropoiesis originates from a different defects. In beta thalassemia, mutations in beta globin genes affects the beta globin synthesis. So the cells, the erythroid cells, are still able to synthesize alpha globin, but without beta globin, alpha globin become insoluble. It interacts with heme. Alpha globin form and heme form aggregates named hemichromes that are toxic for the erythroid cells for two different reasons. First, these aggregates increase oxidative stress by increasing reactive oxygen species. This increased oxidative stress impaired the metabolic fitness of the erythroid cells and in particular affect the mitochondria that is crucial in erythroid cells proliferation and differentiation. It also activates TGF-beta signaling, that is a signaling pathway that counteracts erythroid cells differentiation. On the other side, these aggregates sequester the chaperone HSP70. And this chaperone is crucial since the chaperone protects GATA1 from the cleavage by caspase-3. This cleavage by caspase-3 inactivates GATA1, and this is just to remind you that GATA1 is the main transcription factor that regulates erythroid cell proliferation and differentiation. So without HSP70, GATA1 gets cleaved, and we have increased apoptosis and reduced differentiation of erythroid cells, along with increased proliferation because of the anemia that stimulates hypoproduction. So ineffective erythropoiesis originates. This causes iron overload, the appearance of non-transferrin bound iron that targets many organs, among them also the erythroid cells. And a further accumulation of free iron into the erythroid cells worsen the production of these alpha-globin aggregates and the increase in reactive oxygen species. So, we have learned that basically iron overload is not only the consequent effect of ineffective erythropoiesis, but can also further impair the erythropoiesis in conditions of iron-loading anemia. So one of the main questions in the field is, can we target the hepcidin ferroportin function in order to decrease the iron burden in conditions of ineffective erythropoiesis and improved also erythropoiesis? And to answer this question, first of all, we should know how does hepcidin is regulated in the hepatocytes. So I don't want to go too much into detail, but I should mention that the main signaling pathway that regulates hepcidin expression in hepatocytes is the BMP-SMAD pathway. And the activation of this pathway requires the presence of several proteins. I don't want to go too much into detail. I just want to mention the ligands, BMP2 and BMP6, the receptors, and the BMP co-receptor hemojuvelin. So in the presence of this multi-protein complex on the cell surface, the BMP-SMAD signaling gets activated, hepcidin expression is increased, it is released in the circulation, where hepcidin blocks ferroportin function and reduced iron entry into the circulation. This signaling pathway can be efficiently inhibited by the serine protease TMPRSS6 or matriptase 2 that downregulate the pathway by cleaving hemojuvelin on the cell surface. So when TMPRSS6 is active, the BMP-SMAD signaling is reduced, less hepcidin is produced, ferroportin is stabilized on the cell surface, and iron can enter into the circulation. Interestingly, this mechanism is active also in conditions of ineffective erythropoiesis. And ineffective erythropoiesis by means of erythroferron, FGL1, and PDGF-BB interacts and blocks the activity of the BMP-SMAD pathway, thus reducing hepcidin production, increasing ferroportin stabilization, and causing iron burden. So basically, we can decide to target the hepcidin ferroportin axis by working on 3 different steps in this pathway. By targeting directly the BMP-SMAD pathway, by targeting hepcidin, or, there's something wrong here, by targeting ferroportin. So starting from the first one. How we can target the BMP-SMAD pathway. In conditions of ineffective erythropoiesis, we want to increase the BMP-SMAD pathway. Since the main inhibitor of the BMP SMAD pathway is the serine protease TMPRSS6, we want to inactivate TMPRSS6 activity. There are several papers, and we have shown that in preclinical models, so in animal models, the deletion of TMPRSS6 or the inactivation of TMPRSS6 improve anemia and iron overload in beta thalassemia mouse model. So how we can target TMPRSS6 in order to be able to translate this approach to patients? We can use several anti-TMPRSS6 agents, such as antisense oligonucleotides or siRNA that downregulate TMPRSS6 expression selectively in the liver, or monoclonal antibody that block TMPRSS6 function, with the final aim to increase hepcidin, reduce iron entry into the circulation, and of course, improve iron overload in beta thalassemia. So for sake of time, I will just focus on the clinical trials that have been now published in clinicaltrial.gov and have been terminated or completed. This is the list of compounds that have been tested, of course, in phase 1 clinical trial and in phase 1 or phase 2 clinical trials in beta thalassemia or beta thalassemia plus MDS patients. We have compounds based on antisense oligonucleotides or siRNA, whose mechanism of action is more or less the same. So they aim to downregulate TMPRSS6 expression in the liver. Or we have compounds made by monoclonal antibodies that block the function of TMPRSS6. As you can see, Silence Therapeutics has completed the phase 1 trial on thalassemia and MDS patients. However, they have not released the results. We have only one poster that has been published by Ionis that terminated the phase 2 clinical trial on beta thalassemia patients. And unfortunately, the results seems not to be so encouraging, since they claim that the study was terminated for lack of efficacy. What does it mean, lack of efficacy? The primary endpoint of Ionis was an increase of 1 gram of hemoglobin after 27 weeks of treatment, and this primary endpoint was not reached. This is the reason why they decided to terminate and stop the phase 2 clinical trial. So only from these results, we don't know what are the data from Silence Therapeutics and from Regeneron that are testing the monoclonal antibody and the siRNA. But from these data, it seems that targeting TMPRSS6 in beta thalassemia patient is not effective as demonstrated in the murine models, meaning that we are losing some information in the murine models that properly mimic what is happening in the patients. What about compounds that mimic hepcidin activity? And for this reason, several hepcidin mimetics have been proposed. I want just to mention that one of the main problems in selecting and synthesizing hepcidin mimetics is to increase the half-life of hepcidin, that in vivo endogenous hepcidin has a very short half-life of about 3 hours. So it's unfeasible to generate a compound that fully mimics human hepcidin because the half-life is very low. This is the reason why several hepcidin mimetics have been developed, and I would like to focus on 2 of them, LJPC401 and PTG300 or rusfertide. So LJPC401 have been tested in hemochromatosis. The trial has been completed, but the results have not been published. I forgot to mention that hemochromatosis is a genetic disorder that, as beta thalassemia, it relies on a downregulation, a reduced production of hepcidin and iron overload. Of course, in hemochromatosis, the genetic cause of the disease is completely different. But at the end of the day, both hemochromatosis and beta thals have both a reduced hepcidin and iron burden and iron overload. Also, for transfusion-dependent beta thalassemia, this compound has been tested. The trial has been terminated, so not completed, but terminated. But La Jolla Pharmaceutical did not release any note about the results of the trial. We have been more lucky with rusfertide, so protagonist. They have completed a phase 2 trial on hemochromatosis that have been published in a high-impact factor journal. And interestingly, this compound have been shown to be able to strongly reduce the phlebotomy that are needed to hemochromatosis patients in order to remove iron overload. Now the trial, they have performed also a phase 2 clinical trial on non-transfusion and transfusion-dependent beta thalassemia that have been completed, but the results have not been released yet. So we are pretty much interested to know whether this compound, this hepcidin agonist is effective in blocking iron overload and ameliorating anemia in non-transfusion and also transfusion-dependent thalassemia. And then the last compound is VIT-2763, which is a compound that blocks ferroportin function. This is because due to hepcidin downregulation, ferroportin is stabilized on the cell surface. The compound has been produced by Vifor Pharmaceutical and have been tested in beta thalassemia patients and in sickle cell disease, both in phase 2 clinical trials. We don't have the full results here for beta thalassemia. I just found a report that had been published in Hemasphere in 2022. This is an abstract that had been submitted at the HIA Congress in 2022. And they reported that VIT-2763 is effective in reducing circulating iron in beta thalassemia patients. Unfortunately, they do not describe hemoglobin, but as you can see, the trial lasts only for 12 weeks. For probably the time is not sufficient to see a beneficial effect on erythropoiesis. So to conclude, I hope I have convinced you that a balanced coordination of iron metabolism with erythropoietic activity is a protective mechanism against iron overload and anemia, that the regulation of the hepcidin ferroportin axis is the main cause of iron overload in anemias characterized by ineffective erythropoiesis, that erythroid cells regulate iron homeostasis by secreting erythroferron under condition of expanded erythropoiesis. However, there are other organs and tissue that may contribute to hepcidin suppression in hypoxia, as, for example, PDGF-BB and FGL1. And finally, that the common pathogenetic mechanism of iron-loading anemias allow the identification of several therapeutic targets worth testing as single or combined therapies. And with that, I would like to acknowledge the group in Milan and all of you for your attention.
Thank you very much, Laura. So the chat is open if you have any question for Laura. Of course, I have a question. First question. I don't know if it's a stupid question, but do you think, in my experience in medicine, so what I saw often that finally we use different drugs in the same time. So do you think that finally we will work that way?
Yeah,
for example,
for sure.
that? Okay.
Yeah, for sure. Also because I mean, I focus my talk, and in the last part I describe only drugs that targets the hepcidin ferroportin axis. But of course, the main genetic defects still remain. And in the case of thalassemia, you have a beta thalassemia, beta globin mutations, and an imbalance between the beta and alpha thalassemia that causes several pathological mechanisms. My idea is that for sure the combination of different therapies, one, for example, targeting specifically erythropoiesis, and I'm thinking about the TGF-beta pathway and, for example, activin ligand trap such as luspatercept. And on the other side, targeting the hepcidin ferroportin function can somehow improve the beneficial effect of both these drugs in ameliorating erythropoiesis and iron overload. I'm pretty convinced that we should move towards a combination therapy.
Okay. Thank you very much. And then maybe in totally another field, do you think that there is still a place to use hepcidin as a diagnostic approach? And what do you think about it?
Okay, so I think that for sure there are some forms of iron refractory iron deficiency anemia in which it is crucial to understand whether hepcidin is inappropriately high. I'm thinking about some cases of iron deficiency anemia that are refractory to oral iron administration. I'm not a clinician, but I think that you as clinician know that there could be many causes of iron refractoriness, dietary iron refractoriness, so the inability of a pill of iron to improve iron deficiency in a conditions of iron deficiency anemia. And for example, in this case, having knowing whether hepcidin is inappropriately high or is low as expected in a conditions of iron deficiency anemia can help to drive and to design a better therapeutic approach to the patients. Of course, if in a conditions of iron deficiency anemia, hepcidin is inappropriately high, it doesn't make sense to give oral iron administration to the patients, because in conditions of high hepcidin, I'm expected that the dietary absorption will be impaired. It would be more useful to give a parenteral administration on iron in order to partially bypass the blockage of ferroportin by hepcidin. The opposite, of course, if hepcidin is appropriately downregulated, as in iron deficiency anemia. In this case, a defective dietary iron absorption could be due to many other causes and not to a blockage of ferroportin by hepcidin. And the same, I think, could be applied also to other genetic diseases. For example, also in beta thalassemia. Beta thalassemia, if you want to know whether the patient is able, once, for example, treated with luspatercept, so activin ligand traps that improve erythropoiesis, if you want to know the patient is responding correctly to an improved erythropoiesis, you would expect an increase in its hepcidin levels that can somehow contribute to counteract further iron overload.
Thank you very much. So I don't know if so I don't see any other questions. So I would like to really thank you.
Thank you very much for inviting me
and,
to this exciting
the picture were really
webinar.
I'm sure that anyone who will go on our website will be happy to listen again to the talk. So thank you very much.
Thank you, Beatrice. Thank you very much.